Hey guys, welcome to CakeWalk Cambridge. I'm Aisha and today we're going to start with Chapter 4, Biological Molecules. In my textbook, this chapter was like 4 or 5 pages, and I assure you this won't take much time today. It's a small chapter and it's relatively quite easy, so don't worry about it. Okay, so first we have three food components: carbohydrates, fats, and proteins. You are required to know the elements that make up each of these molecules. So if you have a molecule of carbohydrates, the basic elements present in a carbohydrate are carbon, oxygen, and hydrogen. Also, another tip here for chemistry: when you are asked to define a carbohydrate, you need to say a carbohydrate is a molecule that comprises carbon, oxygen, and hydrogen only. You get a mark for stating only, so never leave it out. Then you have fats, which also comprise carbon, oxygen, and hydrogen. And then you have proteins, which have carbon, oxygen, hydrogen, nitrogen, and sometimes sulfur or phosphorus. Um, it's not a question that is, I've never really seen it in many papers. The different components, like such as phosphorus and sulfur, usually it's these four, but it's always good to know that sulfur and phosphorus also sometimes make up proteins. So you memorize this table and your initial concept of biomolecules is clear. Okay, so larger molecules are made from smaller molecules, as we saw before. Large carbohydrates are made from oxygen, hydrogen, and carbon. So these are some examples that you have to know for Cambridge for sure. So starch and glycogen are made from glucose. So starch is what is stored in the leaf after glucose is formed via photosynthesis. So carbon dioxide plus water in the presence of sunlight and chlorophyll gives you glucose and oxygen and energy. So the glucose is converted to starch, which is a compound that is stored in a plant cell. So starch is made from glucose. Glycogen is another converted form of glucose that is used as storage in humans, in animal cells. It's stored in the liver. So you will learn about this a little later, but you need to know starch and glycogen are made from glucose. Cellulose is also made from glucose. As we discovered in chapter 2, cellulose makes up the plant cell wall. So never forget that. Cellulose is directly related to two things, making cell walls and made from glucose. Then we have proteins, which are made from amino acids. This is a very basic thing that you should have learned in 8th grade. Okay. And then we have fats and oils, also referred to as lipids, which are made from fatty acids and glycerol. You need to know these broken down components, not only for bio, but also for chem. And that's why I stress on the importance of knowing this slide, because all of these are important. Okay, now we have testing different food components. As you can see, you have starch, proteins, reducing sugars, which could also be simple substances like starch broken down into sugars, or reducing sugars that are present in fruits, their pulp, juices. Then you have fats and lipids. And then you have vitamin C, which is present in citrus fruits like orange, lemons. It has ascorbic acid, like those kind of fruits. Okay, so for starch, you have a very easy chemical test. You add it to a solution of iodine. If the food sample contains starch, it will turn into a blue-black color. If not, the color of the iodine solution remains yellow-brown. This is important also to test for the storage of starch in plants, but there's a different method. So you don't use the same method for plants. So in bio, when you are asked to test for starch and only starch, like from just granules of starch, you don't use the plant test. You just simply add it to iodine solution. And also for this experiment, to make your color change easier, you use a white tile. You carry it out on a white tile so that the color change is more visible because there's a greater contrast. Then we have proteins, which the chemical test is the biuret solution. You should know the name biuret solution, and you should also know what it actually is, which is basically sodium hydroxide followed by copper sulfate. You don't need to know the concentration or volume of each. You just need to know the two solutions that are added to the food test. If there's a positive test result, there's a color change from blue to violet. And if not, the blue color remains. This table, there's a lot of mugging up, but you get so many marks for ATP just to remember this. It's completely worth it. So just go for it. Then you have reducing sugars, which is very frequently asked for like 5 marks, where they first ask you what the chemical test is. That is, you add Benedict's solution or Benedict's reagent, which is a solution. You don't need to know what it comprises. It's a solution. And you heat it to 80 degrees Celsius. You do that using a water bath. A water bath is basically when you have a larger container or beaker filled with water at a certain temperature, say 80 degrees Celsius after being heated, and then you add the test tube with the Benedict solution and the reducing sugar inside it. So the heat from the water is absorbed by the test tube and it transfers to the molecules inside. A positive test, then you also, that's like your first 2 to 3 marks that you get for the question. Then you get a mark for stating the positive test result. Always remember to state the color change. Don't just state orange or brick red. State blue to orange. Okay. It's very interesting to note that there are a variety of color changes seen here: blue to green to yellow to orange to brick red. You need to know that green, so blue has 0 concentration of reducing sugars, green has slight concentration of reducing sugars, yellow has a greater concentration, orange has a moderately large concentration, and brick red has a massive concentration of reducing sugars. So if you know the color, you will know the concentration of reducing sugar present in the solution. And then again, the negative test result is that the blue color remains. Then you have fats and lipids, where you use the ethanol emulsion test. Okay, just one more thing to add to the reducing sugars, you also get a mark for stating how you could safely carry out the test. That is because you're using heat, use a water bath, gloves, goggles. Always. Goggles and gloves are a must to write, especially goggles. You always get your mark for safety there. Then you have fats and lipids, where you use the ethanol emulsion test. It's very important to know the name, ethanol emulsion test. You get a mark for it. The components of it are ethanol and water, and you get a cloudy white emulsion, like a cloudy white color if the test result is positive, if fats are present, and if not, it's colorless. Then you have vitamin C, which you use the DCPIP test, which is like, so if you add DCPIP to vitamin C, that is in lemon juice, there's a color change from blue to colorless, and if it's not, it's just simply blue. So for these tests, you need to remember you get a lot of ATP questions based on them. That is planning an experiment, which we will also have a separate video for. Then you have the first initial experiment given in your ATP paper as a question. You will have questions based on that. You will be asked how to carry out a test. You will be asked how to safely carry out a test, and you will be asked the positive test results. Negative test results I've not really seen, but you obviously have to know it. It's part of your syllabus. Okay, so proteins. Proteins are made of amino acids, as mentioned earlier. Different sequences of amino acids give different shapes to protein molecules. So this is because protein molecules are specific, which means each of them have a unique structure that is suited to their function. So different amino acids, different sequences of amino acids give rise to different proteins, which have different structures, which have different functions. That's basically how it works. So this last point here about protein molecules is taken from the mark scheme of a paper where they asked me the role of proteins, and I found this question interesting. So I thought I'll pen it down for you guys. So proteins are used in A, carrier proteins, which we discovered while studying active transport. Carrier proteins are used to transport molecules from a region of lower to higher concentration using energy from respiration. Enzymes, which you will be learning in the next chapter, they catalyze reactions. Cell membranes, proteins and lipids form cell membranes, which is the boundary around the cell. Hemoglobin, which is in red blood cells, and they make a cytoplasm. As stated earlier, cytoplasms, which is the watery jelly-like structure in a cell, contains lipids and proteins in its area. That's what the cytoplasm is. So let's move further. Yeah, so this is a part taken from an MCQ paper. You have the structure. So if you have an enzyme, it's always going to have an active site that we will learn in the next chapter. If you have an antibody, it will always have a binding site, which you will learn in chapter 10, don't worry. And when you have DNA, you have the base order. So this is basically what you need to know for now, because in MCQs you're often asked to match the correct combination together. So these are the correct combinations that I've seen over a variety of papers. So learn this by heart and you'll be good to go. Okay, DNA, very, very important concept. So a DNA has, so this is basically the structure of DNA. It has two strands coiled together to form a double helix. Each strand contains chemicals and bases. Cross-links are formed between the strands by pairs of bases. A pairs with T, C pairs with G. Okay, so let me explain this to you right now. So this is basically the structure of DNA. A double helix with a coil. So it's going to be something like this, because there are two strands. Okay, forgive my drawing. Okay, then you have different bases on each segment of the strand. Okay, so if you have the base, a nucleotide A, G, and C. Okay, supposing. Now you need to understand, so A is basically adenine, T is thymine, here T is thymine, C is cytosine, and G is guanine. So A pairs with T. You do not need to know the names. You just need to remember A pairs with T and C pairs with G. So if you have A here on one side, you'll have T here on the other side. If you have G on one side, you have C on this side. If you have C on one side, you have G on the other side. They're always complementary pairs. Okay, A with T, C with G. So once there was an MCQ where they asked what is true, which statement is true, and one statement was A always pairs with T. You tick that because that is true. A always pairs with T, always being the key word here. Then DNA is basically used for genes to code for proteins which control cell functions. This has also been taken from a past paper mark scheme where they asked you where they asked me what the role of DNA was. So one is to code for proteins, used for genes, and to control cell functions. Water. Water is a solvent, okay? As everyone knows, if you've probably studied this in chem, it's a universal solvent because it has the capacity to dissolve a wide variety of substances. Water is used as a solvent in digestion, excretion, and transport. You will learn about this later as well. So water also forms part of the plasma. Water is used in the kidneys. Water is reabsorbed. There are like so many functions of water, but the main primary thing you need to know is solvent, and then digestion, excretion, and transport. 60% of the body. Okay, you don't really need to know this. I thought I could just include a fact. This is just an approximate number. You don't have to keep it in mind at all. And there's another point that was mentioned in my textbook. I've never really seen it in mark schemes, but I found it intriguing. It dilutes toxicity. So if you have a large amount of water and a small amount of a toxic substance, a waste product from metabolism, its toxicity is obviously going to be dialed down because water has a diluting effect. So that was water. Okay, everyone, so this was biomolecules, just a like 13-minute long video. I hope you enjoyed it. I hope it helped you. I'm not really sure. If you have any doubts, please feel free to email me. I'm always there for you guys. So please like this video, subscribe to CakeWalk Cambridge, share this video, and turn on post notifications so we can get through this journey together. Bye!